So yeah, I'll talk about HippoMaps. First of all, I want to say this project was heavily inspired by NeuroMaps, which you guys will hear more about tomorrow. I won't go into much detail, but basically this is a huge collection of different kinds of data mapped onto neocortical surfaces. And this is awesome because we can do a lot of cool things. So we could take a new individual and try and fingerprint them with respect to a bunch of different maps like this and try and determine if they're an outlier in some sense. Or on another hand, something we could do is examine the relationships between maps to see which ones are correlated and if that has some kind of functional significance or theoretical significance. So one of the key aspects of this project, though, is that we can register between different brains. So in the neocortex, we typically do this by inflating to a sphere and rolling these spheres against each other to maximize their overlap. So this is very precise and probably the most common method to register different individuals in detail. In the hippocampus, of course, things are a little different. But I would argue not too different. So a lot of people refer to the hippocampus as subcortical, but it's actually cortical. It's archicortex or old cortex. Those familiar with my work, I spent a long time on this problem of the geometry of the hippocampus and understanding how we can do similar operations as the neocortex. In particular, we can map out this sort of this curved long axis and this medial lateral, or if you like, proximal to distal axis of the hippocampus in order to unfold it. And so basically, in the end, what we have is a pipeline where we can take two different individuals. So one example on the left and one example on the right. So the one on the right is BigBrain. And we can actually, even though they have different morphometry and different numbers of folds and sulci and gyri within them, we can map them first to a surface and then to a flattened surface. And then even though these flattened surfaces are not perfectly registered, we actually just have a new paper that should be appearing any day in eLife that we have really fine scale mapping now using this unfolded space. So it's really equivalent to doing a surface registration of neocortical data. So with this tool, I basically want to do something like NeuroMaps. So I want to come up with a common space repository for multimodal and multi-scale hippocampal mapping. And of course, this will include a lot of the cool things about NeuroMaps, like really nice tools and tutorials and robust methods. So here's just a quick overview of how this works. So here on the left, I have a few different slices from a histological data set. And basically, you can see I've fit a few surfaces to the hippocampus here. And then in the middle, I have this 3D reconstruction of a hippocampal mid-thickness surface. And then on the far right here, I'm showing just sort of a canonical folded and unfolded surface next to each other. And the key point to understand here is that even though a lot of individuals have different shaped hippocampi, they can all be mapped to this common space. Of course, when we do a lot of mapping, we always run into problems. So we have things like misalignments or slicing artifacts or missing data, depending on the source of the data. So I just wanted to mention a little bit about how we keep this mapping technique robust. So one of the nice things about surface-based representation is sometimes we can actually have a pretty good way to fill in missing data if it's just a piece of the tissue that's missing. And we can also do things like sample immediately adjacent to the surface. So if you look at the top left, I have the orange and the red are the inner and outer hippocampal surfaces, but then I also extrapolated them a bit further in the green and red. And this allows us to then sample data along the columnar direction of the hippocampus that's actually outside of the hippocampus. And so if we have misregistered data, we can still include that into our unfolded mapping of the hippocampus, and then we just do some alignment of that data to hopefully get really nice columnar organization. And then I'll show just a few maps from the mid-thickness. So the maps I'm going to show are going to follow this canonical folded and unfolded space. So I'm going to leave that in the top corner. And here I just wanted to show the BigBrain Merker staining. So this is average across the left and right. And of course, it has a lot of nice properties. So there's way more resolution available, as Alejandro was talking about earlier today. And so there's a lot of cool stuff that we can do with that. But I want to, in this project, go really wide and talk about integrating lots of data from different sources. But one thing to note about this is we see mainly these vertical bands in the data. And the vertical bands, if you look at the top right, this is actually the different subfields of the hippocampus. So I just wanted to highlight, you'll probably see a lot of this vertical banding, and that's because the subfields is a major structural difference inside of the hippocampus. So as I said, I'm taking data from a lot of sources. I'm not going to go into detail about all of them, but I'll just basically say it's spanning from microscale histology into fMRI at the scale of about 2 millimeters using in vivo, ex vivo scanning, 3T, 7T, 9.4T, lots of cool methods and open source data that are very valuable. This was back to BigBrain, and I'll just swap out the data now. So same format here, but this is now the transmittance from a methodology called polarized light imaging that Marcus Axer and group are working with. So this is very cool because we can now map out things like neural processes within the hippocampal tissue, or within the gray matter of the hippocampus, I should say. And we can additionally access lots of cool information about the PLI that I won't get into in too much detail right now. From another data set, so this is called the ahead brain, we actually have other kinds of stains as well. So this is a Bielszowski stain for nerve fibers, and you can see it's distributed very differently across the hippocampal surface. But again, we see more or less these vertical bands that correspond to the subfields. Just another example here, I won't go through all of them, but this is an ex vivo MRI at 9.4T. So this is 1 over T1, basically. So if you are doing imaging, it's very related to the T1. And so there's very common contrast in MRI. And here we're mapping this at a very fine spatial scale. But we can also then look in vivo MRI. So this is quantitative T1 now. And we can see it's anti-correlated with 1 over R1, as we'd expect. So this is a really common contrast, and it gives us some information about intracortical myelin. So that is the amount of myelin within the cortex itself. And again, it follows this vertical banding. We can also look at things like diffusion-weighted imaging. So this is a measure called the apparent diffusivity coefficient, which is basically getting at the amount of water versus the amount of fat within a voxel. And then I want to just show a few more examples. So this is some resting state functional MRI data where we've mapped a feature called intrinsic timescale, which is basically how much time it takes for the autocorrelation to decay. So it's really thought to be indicative of recurrent connections within a voxel. And again, we see this sort of vertical banding, and now I'm showing the left and the right hippocampus, and you can see again, it's highest in the medial parts of the hippocampus. So this would correspond to the subiculum subfield if you look at the map at the top. And then we can then look at functional connectivity. So here's just the average functional connectivity with any part of the neocortex. But we can take this data and we can actually then decompose it and ask, well, which parts of the neocortex are we interested in? So when we do that, we use a method called diffusion map embedding through this brain space tool. And we can see that the primary gradient that explains variance in where the connectivity is actually, it doesn't follow the subfields at all. It doesn't show this vertical banding. Instead, what we see is a very strong anterior to posterior difference. So in other words, the bright colors are all connected to one set of neocortical areas, and the darker colors are all connected to a different set of neocortical regions. And this is in agreement with some previous literature as well. So with all these features in mind, we can then examine also the relationships between features. So here I've put a box around each method that uses the same source of data. So we can look at whether they're correlated, anti-correlated, these sorts of things. And then we can also take that data and do cool dimensionality reduction methods. So here I'm just showing the first and second principal components. And you can see I've colored the dots here according to which subfield they come from. And so you can see these two principal components, they really actually nicely separate out the subfields of the hippocampus, which makes sense because we know that they're one of the primary structural factors of the organization of the hippocampus. The other way to think of that is the subfields follow this proximal distal, or in other words, medial to lateral organization. So again, we see that if we just color them by their medial lateral or proximal distal, if you like, location. But they don't really follow an anterior-posterior axis, at least not very strongly. So I just want to also just show one more other cool dimensionality reduction technique, which is, again, this gradient decomposition. And so here we have basically gradient 1 is a similar but different version of a PCA component 1, where, again, we see this vertical banding that really each of these components is basically highlighting different subfields, we think, of the hippocampus. So altogether, the hippocampus is archicortical, which means we can unfold it or map it along these two unfolded dimensions. And this is really interesting because the proximal distal axis really seems to be related to, of course, the subfields, which we see through lots of types of data, so histology, quantitative MRI and diffusion-weighted imaging, as well as even local fMRI signal properties. However, the global fMRI signal properties, so that is the functional connectivity to the rest of the brain, is totally different. It follows this perpendicular axis that is anterior to posterior. So to me, this is sort of suggestive that what the hippocampus is doing is across its proximal distal axis performing some set of computations with different inputs and outputs across its anterior posterior axis. With that, I'll just maybe make a comment about the tools that we're developing here. So we're following these FAIR principles. If you're interested, you can follow this project on GitHub. And yeah, so on the left, I'm just showing hippocampi with different spatial scaling. And on the right, this robust hippocampal spin test equivalent to the spin test we do in the neocortex. And of course, I want to thank everybody who's contributed to this project because it comes from a lot of great people and a lot of great data sets. Thanks.
Thank you, Jordan. Thank you. Questions? Alan? I can, hold on.
Great stuff, Jordan. I'm just wondering if you've had any thoughts about looking at Jake Vogel's gene expression maps of hippocampus and seeing how those gene expression maps relate to your...
Yeah, I would like to. It's not something I've gotten to yet, but I think that would be a really useful thing to have in this sort of repository. So that's to say I haven't yet, but I would like to, and we should maybe get started on that.
Quick question from me. For the spatial resolution of the fMRI on the hippocampus, for the functional MRI data, what was the resolution you were able to work with in order to get meaningful results?
Yeah, it was 2 millimeters. And what's cool about doing this surface-based representation is you can actually generate a surface that has more or less vertices that can sort of match the resolution of your data as well.
Very cool. Thank you very much. Yeah.
Thank you.
So.